Good morning, students. Today we discussed about population genetics and evolution. All the individuals of a species constitute a population. The genetical studies for the inheritance of phenotypic traits in a given population is called population genetics. The population genetics is a quantitative science to calculate the results of the mode of inheritance of genes in a given population. Various statistical and mathematical models are employed in it. Certain fundamental aspects of population genetics are the following. First is Mendelian population. A population of a particular species includes many inbreeding groups. The inbreeding groups may form a community within defined geographical boundaries and are called Mendelian population. A Mendelian population, thus, is a group of sexually reproducing organisms with a relatively close degree of genetic relationship, like species, subspecies, breed, variety, strain, etc., residing within defined geographical boundaries where inbreeding occurs. Gene pool and gene frequency. To get a F2 generation 3:1 phenotypic ratio of a monohybrid cross, we begin with two homozygous parental strains such as capital A and small a. That is, we introduce that alleles capital A and small a in equal frequency. But in a Mendelian population, frequencies of alleles may vary considerably. For example, Mendelian population of a man, the gene for the polydactyly is dominant, yet the polydactyly phenotype is fairly infrequent among infants. This indicates that frequency of the dominant allele in population is lower than that of its recessive allele and that both alleles do not exist in population in the 1:1 ratio like the individuals of monohybrid cross. Further, if all the gametes produced by a Mendelian population are considered as hypothetical mixture of genetic units from which the next generation will arise, we have the concept of a gene or gamete pool. The percentages of gametes in the gene pool for a pair of alleles depend upon the genotypic frequency of the parental generation whose gametes form the pool. Thus, if a population is of dominant genotype capital A, then the frequency of dominant alleles in the gene pool will be relatively high and the percentage of gametes bearing the recessive small a allele will be correspondingly low. Two models of gene pool structure. First is classical hypothesis. It was developed by T.H. Morgan in 1932 and supported by H.G. Muller and Kaplan. The classical hypothesis proposes that the gene pool of a population consists of a gene locus of a wild-type allele with a frequency approaching 1. Mutant alleles in very low frequencies may also exist at each locus. A typical individual would be homozygous for the wild-type allele at most gene loci at a very small proportion of its loci, the individual would be heterozygous for a wild and mutant allele, except in progenies of consanguineous matings. Individual homozygous for a mutant allele would be extremely rare. The normal ideal genotype would be an individual homozygous for the wild type allele. According to classical hypothesis, mutant alleles are continuously introduced in the population by mutation pressure but are generally deleterious and thus are more or less gradually removed from the population by natural selection. Periodically, a beneficial mutant allele might arise conferring higher fitness upon its carriers than the pre-existing wild-type allele. This beneficial allele would be gradually increased in frequency by natural selection to become the new wild-type allele, while the former wild-type allele would be eliminated. Evolution thus consists of the replacement at an occasional locus of pre-existing wild-type allele by a new wild-type allele. Second is balance hypothesis. This hypothesis was proposed by Dobbs, Hansky, and E.R. Ford. This hypothesis was derived by direct study of natural populations. According to the balance model, there is generally no single wild type or normal allele. Rather, the gene pool of a population is envisioned as consisting at most loci of an array of alleles in moderate frequency. A typical individual is heterozygous at a large proportion of its gene loci. There is no normal or ideal genotype, only an adaptive norm consisting of an array of genotypes that yield a satisfactory fitness in most environments encountered by the population. The proponents of the balance hypothesis argue that ubiquitous allelic polymorphism are maintained in population by various forms of balancing natural selection. The fitness conferred on is carries by an allele depends on what other alleles exist in the genotype at that and other gene loci. It also depends, of course, on the environment. Gene pools are co-adapted systems. The set of alleles favored at one locus depend on the sets of alleles that exist on the other loci. Next is Hardy-Weinberg law. The formula p plus q square, p plus q whole square is equal to p square plus 2pq plus q square is expressing the genotypic expectations of progeny in terms of gametic or allelic frequencies of the parental gene pool and is originally formulated by a British mathematician Hardy and a German physician Weinberg. Independently, both forwarded the idea called Hardy-Weinberg law equilibrium. After their names, the both gene frequency and genotype frequencies will remain constant from generation to generation in an infinitely large interbreeding population in which mating is at random and no selection, migration, or mutation occur. Should a population initially be disequilibrium, one generation of random mating is sufficient to bring it into genetic equilibrium, and thereafter the population will remain in equilibrium as long as Hardy-Weinberg conditions persist. Hardy-Weinberg law depends on the following kinds of genetic equilibrium for its full attainment. First is the population is infinitely large and mate at random. Second is no selection is operative. Third is the population is closed, no immigration or emigration occur. Fourth is no mutation is operative in alleles. And fifth one is meiosis is normal, so that chance is the only factor operative in gametogenesis. The significance of Hardy-Weinberg equilibrium was not immediately appreciated. A rebirth of biometrical genetics was later brought about with the classical papers of R. A. Fisher beginning in 1918 and those of Sewall Wright beginning in 1920. Under the leadership of these mathematicians, emphasis was placed on the population rather than on the individual or family group, which had previously occupied the attention most Mendelian geneticists. In about 1935, T. Dobzhansky and others started to interrupt and to popularize the mathematical approach for studies genetics and evolutions. Genetic equilibrium. Hardy and Weinberg alleles segregating a population tend to establish an equilibrium with reference to each other. Thus, if two alleles should occur in equal proportion in a large isolated breeding population and neither had a selective or mutational advantage over the other, they would be expected to remain in equal proportion generation after generation. This would be a special case because alleles in natural population seldom, if ever, occur in equal frequencies. They may, however, be expected to maintain their relative frequency, whatever it is, subject only to such factors as chance, natural selection, applications of Hardy-Weinberg law in calculating gene frequencies in a population. The gene frequency for autosomal and sex chromosomal allele can be determined by the help of Hardy-Weinberg law by the following method. Calculation of gene frequencies of autosomal genes. An autosomal gene's locus may have codominant alleles, dominant and recessive alleles, or multiple alleles. If one desires to determine the gene frequencies for each of these kind of autosomal alleles in a given population, he has to adopt the different methods. Factors influencing allele frequency or deviations from Hardy-Weinberg law is the Hardy-Weinberg explanation of equilibrium in the allele frequency pattern of a population required three assumptions. First is individual with each genotype must be reproductively fit as those of any other genotype in the population. Second is the population must consist of a large number of individuals. And third is random mating must occur throughout the population. The Hardy-Weinberg theorem with its assumption does not account for any change in allele frequency within population. That is just what Hardy and Weinberg intended because their formula described the statics of a Mendelian population. The shift or changes in gene frequency can be reduced by a reduction in population size, selection, mutation, chance, meiotic drive, and migration. First is selection. It involves evolution and natural selection. Evolution has been described like a process that converts variation within a population into variation between population, both in space and in the time. The most prominent theory regarding the driving force behind evolution is of course the Darwin-Wallace theory of natural selection proposed in 1858 by C. Darwin and E. Wallace. The natural selection theory holds that as genetic variants arise within the population, the fittest will be at a selective advantage and will be more likely to produce offspring than the rest. As the fit continue To enjoy a greater survival, reproductively new species will eventually evolve. Second is natural selection. Natural selection is generally believed to be the prominent agent for determining the relative frequency of alleles in a population. Natural selection differentiates between phenotypes in a population with respect to their ability to produce offspring. One phenotype may better survive endemic onslaughts of parasites or predators than another. One may penetrate new habitats more effectively than another. One may more efficient than other. One may even prey on the other. The important point is that some natural situations or environmental features selectively allows one organism to develop and to propagate more efficiently, and one genotype is thereby afforded greater representation in the population gene pool. If the selective process continues over many generations, allelic frequency will change significantly and the potential will arise for evolution change further. Allelic frequency will change significantly and the natural selection process, while acting on the total genotype, will in fact influence only the heritable portion of the phenotype. If a trait has a high heritability, selection can rapidly affect its frequency within a proportion, whereas selection will take a great deal longer to have any effect on a trait with a low heritability. Natural selection includes the following three parameters: survival rate, relative fitness, and the selection coefficient. To calculate values for these parameters and thus best understand their meaning, we can consider the data from the particular population. For simplicity, we shall consider only a single gene locus in the population defined by the capital A and small a alleles. We shall assume incomplete dominance so that capital A small a heterozygous can be distinguished phenotypically from capital A and small a homozygous. We then count the number of capital A small a and capital A small a and small a individual in a given generation immediately before and immediately after some selective event, the introduction of a parasite, a change in temperature has occurred, and the representative members have been present. Survival rate of the fitness and selection coefficient. This topic is completed. Now, thank you, students.